Good morning. Good to see you all back here again today. So maybe it was a good day yesterday seeing that you came back. But we're going to start off this morning. We're very pleased to have Boris Markov, who's going to talk to us about the 105-day study. The questions came up about it yesterday, and he's going to discuss it, and we'll also entertain some questions this morning. Boris?
Good morning. В последние годы внимание всего человечества все больше и больше привлекает
In recent years, the attention of
дальний
humanity
космос.
is directed more and
И
more
одно из
towards the
первых
distant
мест
universe.
занимает Марс как объект изучения,
One
как
of the
подобная нам
first
планета,
places to
как
study is
возможная
planet Mars as one
одна
of
из первых
somewhat
целей
similar
для
planets to the
освоения
Earth,
околосолнечного пространства.
places of development of solar system.
Раз, два. Марс привлекает внимание
We are attracted
с
by
точки зрения возможности понять
the capability of understanding of
развитие Солнечной
the
системы,
history of development of
развития жизни,
solar system, the
может быть,
development of
историю
life,
угасания.
and possibly
В последнее время
the history
достигнуты большие
of termination
достижения
of
в
life.
дистанционном зондировании
Lately,
Марса, в изучении
distance
автоматическими средствами поверхности Марса.
diagnostic of Mars
тем не менее,
surface
я думаю,
has been
никогда
taken over.
However,
человек от того, чтобы
I think that people
побывать на
will
этой планете.
never give up the
И
idea
в
of
этом случае
visiting the
эффективность
planet,
изучения,
in which case the
анализ полученных результатов
effectiveness
будут
of the
действительно
research
на более
and
высоком
the analysis of the results are going to be at a much higher level.
уровне. Является главным
Humans
приоритетом
are the
марсианской миссии.
primary priority of the Martian, of an eventual Martian mission. It is the human is also the most.
От него зависит в конечном итоге
The human is also the most vulnerable link in the chain
ее реализации.
in implementing a Martian mission.
Принципиальное отличие
The
от
basic
летального полета
difference of an
от полета
orbital
межпланетного
flight and an interplanetary one
заключается в том,
is
что после
in the fact
ухода с околоземной
that
орбиты
once we leave low Earth orbits,
восполнить какие-либо
we are
ресурсы,
unable to
необходимые
replenish
для
any resources that we
осуществления этой миссии
require
для существования экипажа.
for the subsequent mission and for crew life support.
В орбитальном полете
In an
возможна доставка.
orbital mission, resupply is possible.
Невозможно получение помощи с Земли,
It is
включая
not
возможность
possible either to
возвращения
obtain
по
assistance from the ground, and it is not possible to get back to Earth, which is an additional psychological factor that will affect
в
humans.
орбитальном полете
In a low
функция управления
low Earth orbit flight,
во многом возложена
the
на
control
наземные службы,
function is largely dependent on the control center, on the ground support services, which not only monitor continuously all the systems, but also, in most cases, control the vehicle by commanding it remotely. In an interplanetary mission, these functions will mostly fall to the crew. The crew should be able to manage itself, to control on its own, which will include independence
принятии решений,
with respect to decision making,
самостоятельное разрешение возможных
including independent resolution of possible contingencies,
самоконтроль жизнедеятельности экипажа, в том числе
which will
состояние
also
здоровья,
include
психического состояния,
self-monitoring, including health monitoring and psychological status, as well as performance.
для межпланетного полета
For an interplanetary flight,
изменения коммуникации,
what you would find
связанные как с уменьшением объема коммуникации,
is a change in communication, reducing the volume and experiencing a lag in data transmission, which in a Martian orbit or closer into Mars could be as long as 30 minutes, in which case voice communications are not possible, and the only interchange that's possible is an electronic interchange between the crew and ground support. And landing a manned vehicle on an unknown planet is something that's completely new and previously unexplored by manned spaceflight, as is operation on board on the surface of the planet and in the Martian orbit. The crew will find it very difficult because the crew remaining in orbit will have to work much harder and cover for the rest of the crew which are on the ground, and they will also need to coordinate with the ground, with the controllers back on Earth. Essentially, these are the unique features of a Martian mission that we concentrate on in the Mars 500 project. This is a project that started 4 years ago. Initially, it needed to be justified and rationale needed to be provided for engaging in a project like that. At that particular moment in time, we needed to justify it to Roscosmos. We were able to do that, and Roscosmos is the primary customer for this activity. Subsequently, we needed to upgrade our ground medical and engineering facilities. We needed to establish new modules. Right now, this is a set of 5 modules that includes a habitation module, which is the primary location where the crew is going to be spending most of their time. There is a medical and research module. There is a storage module, although it is not purely storage, because in addition to housing all of the foodstuffs and all of the other resources required for the crew to sustain themselves, we have a greenhouse as well as a workout room and some medical equipment. This set of modules also includes a module which is designed for a Mars landing, and the final component, which is part of the vehicle, is a module that simulates the Martian surface, which has been introduced recently. I can talk to you of the engineering properties of the vehicle if you're interested in more detail. The crew that is currently isolated on the simulated vehicle is a crew for a 105-day experiment, and we will not be able to simulate a complete Martian mission, of course, from the duration standpoint. And our concept is that we are going to simulate a target-oriented ISS flight of 105 days, which reproduces some peculiarities of the Martian mission. This experiment was preceded by two verification experiments of 14 days each, which mostly verified the life support system for the crew. Every time we introduced a new module, the vehicle configuration changed, and we needed to verify the new configuration like this. We split the 105-day mission into 3 series of 35 days each. And each of the series, in each of the series, we're attempting to keep to the following constraints. Stringent isolation, which is mostly related to the fact that the crew has no communications capability, which are available currently to the ISS crew, and that was previously available to our test subjects in similar experiments of even greater duration. Like, for example, there is no way to use a phone. The crew does not have internet access. Limitations have been imposed on interchanges with the support team. We have introduced a rule that the crew will initiate communications, and the support team is supposed to respond or provide a tag up or provide information. The crew will be the lead in setting up interchanges with the support team. All of the resources required for crew life support are already on board the simulated vehicle, including mostly food, clothing, life support components, sanitation and personal hygiene items, things related to medical support and medical treatments. Everything is inside the simulated vehicle, and the crew is supposed to use these resources on their own. And this is a test, as far as we're concerned, of the correctness of the utilization standards for groups and individuals for this period of time. Their independence is also supported by the fact that the crew is not able The crew is expected to find resolutions to any situations that arise in the course of the experiment. They do not have any access to technical or medical assistance from the ground, which can be provided, but under exceptional circumstances when there is risk to human health or risk to some equipment that's unrecoverable, which may arise in the course of the experiment. The final 35-day stretch is going to be characterized by the communications mode, which is expected in a Martian orbit. and which will probably be confined to 2 or 3 packet messages from the crew to the support team and back from the support team to the crew. One other objective of this final stretch is introducing the module that simulates a Martian lander, such that the crew is able to verify all of its systems in an integrated manner together with the primary modules for the crew to evaluate its human engineering properties and for the crew to perform a number of activities which will be novel for the crew and which the crew is going to be notified of electronically in the course of the experiment. After launch, that is. We talked about the selection and the training for the crew which we had. I will reiterate through that very briefly. The crew is a 6-person crew, 4 Russian crew members, and 2 European Space Agency crew members. The crew has the following functional positions. We have a crew commander, we have a flight engineer, we have a crew surgeon, and we have 3 researchers. The experiment has a very heavy research program. There are over 70 projects that are being implemented in the course of the experiment, mostly, as expected, psychological investigations. And the objective in the training was to distribute the responsibility for these experiments out to the crew members, and we have defined clearly who is responsible for each specific experiment, who is assisting, and who is supposed to be monitoring the state of health if required. But in the planning of these activities, the crew was heavily involved because they had the initiative and we just assisted in apportioning the responsibilities in a rational manner, in an efficient manner. It should be noted that most of the investigations are multifaceted investigations, and it is difficult for a psychological experiment to find someone that would be happy with just a questionnaire. There is some recording of physiological parameters, some biochemical parameters, which may provide insight into the condition of the body and the psychological and emotional stress. So these are integrated investigations, and we had to combine research proposals in certain instances to avoid duplication and save crew time. At this moment in time, we are close to the midpoint in the experiment, and this transition to standalone operations was very difficult for both the crew and the investigators, because frequently both the crew members required or requested things that we did not want them to request, or we didn't want to provide in the way of additional information or resources. On the other hand, when investigators through video, because it's very well provisioned with video, when the researchers see that the crews are not doing the right thing, the researchers are attempting to intervene, and they're saying that on station we're able to contact them and tell them to adjust their activities, but we have to hold them back because the crew is out there somewhere away from the Earth, which means that he or she has to be on their own, and you can correct the procedure once you get the results back. And so over a month we have minimized the activity on either side. We were lucky because we have a very good crew, and I think that we should mention the professional qualities which have been demonstrated by members of the crew, and we have to speak about their personal character traits that we wanted and we found. In addition to being healthy and professional, they have to be very creative for an experiment like that, and for an interplanetary mission even more so. They need the ability to digest new information, to learn as they go along, because very frequently procedures change, new objectives arise, and crew members have to play off their sheet of music without having practiced. Motivation is very important. Motivation coupled with conviction that a contribution, personal contribution, is required to succeed. It is also very important that the person be critical towards his or her conduct. They need to be responsible. They need to take on responsibility for the consequences of their actions. Also, it is very important that they be able to get along with the rest of the crew. They need to measure their actions and their emotions by the actions and the emotions of the rest of the crew, and if required, be able to subjugate their requirements and their desires to the overall objective. We would like to see them tolerant to one another. We would like to see them behaving naturally, because it's difficult to play a part for a very long time. So in a critical situation, a person is always him or herself. We also require a non-aggressive sense of humor. We have two cosmonauts. One is a researcher and a commander working at our institute, and one flight engineer is an Energia person. We have a practicing physician on the crew, a very good one. We have a physical exercise specialist on the crew as well. And as far as our European colleagues are concerned, they do a very good job, and they fit in well with the crew. One is a civilian pilot, a commanding officer of an Airbus 320, Cyril Fournier, a French person. The other is an engineering officer that has some experience in Afghanistan.
Надо сказать, что прекрасно и ответственно работают люди.
And they're working very well, they're very responsible, and we're very happy
Наша главная задача теперь получить
with the way they're doing, and the primary objective
экипаж для основного
right now is to get together a crew for the 520-day experiment, which would mimic the current crew that we have in its properties. And I would like to say right off the bat that we are writing a program for the 520-day experiment with a little past experience with the crew in this current experiment. We have announced, we have made a call for candidates for this crew. The European Space Agency is also working to provide us with some candidates. It is very important to have an overall concept for this mission. The changes in the communications which we introduced in the 105-day experiment overnight after day 70, which is what we're going to do. We expect to do gradually in the longer experiment at a time when you would expect this to occur in a real Martian mission, where this delay lengthens over time. And it is quite clear that the first 70 days, the delay is not going to be significant and will not affect the crew to any serious degree. However, it will grow exponentially after that point in time, and it will grow in importance. It is a major problem for us to apportion the various functions to the crew members, to identify the crew members that are going to land on the surface of Mars. We would like to get a virtual model of the Mars landing.
Вы знаете, что вот у нас есть такое и на станции.
You know that we do have this in our experiment and on the station both. This is a research program which is called the Pilot. And here we investigate the skills of a pilot, which is for docking and undocking operations. Also in some complex environment. We would like to get a virtual model of such dynamic operations for landing on Mars surface. Activities on Mars surface is actually an ongoing project, and we do have some activities done by Moscow Aviation Institute in preparation for that. Also, the Moscow State University is involved. They do have the virtual model for Mars surface already, and they have a proposal to use prototypes of Martian rovers. Also, it is possible that probing of landing site can be done by using flying means like aerostats and other vehicles. Because the crew activity, it was mentioned yesterday very well, about the activities within models for Mars. I mean, you cannot walk away far. You need to know which direction to take, and you need to know what exactly you are looking for. That's why it is important to start at least to develop a system that will be used automated means, where
a
person will be supporting such systems, will be getting information and analyzing the situation, and later on this data can be used for some specific paths where a man or woman would take part already. And later on some sampling can be taken of the ground, of other items. And then we will already know which place they will be investigating and where exactly they will be taking the data from. Of course, this is a huge project. It is good that we have some room for creativity here. On the other hand, we have a deficit of time. And one experiment is ongoing right now. The other one is going to be started in September. In September, we are going to start working with the group. Right now, we are in the preparation phase. And in the beginning of next year, we'll start the experiment with 520 days duration. We are convinced that we are on a good schedule and what we are doing is timely. I think this is just a principle that we adhere to at our institute. Prior to jumping into a huge project, we have to do preparation work on the ground. This is what we were doing prior to long-duration flight. We had a 970 bed rest experiment in the past. At that time, we were developing countermeasures not only for long-duration flights, but also for Martian projects. Those countermeasures that were developed at that time can be applicable. Also, prior to introducing females to space, we had a bed rest experiment using a female subject. In 1967, long time ago, it was one of the first unique confinement isolation experiment where we used biological systems of life support. Such ground preparation work will give you the initial base data to move along. Also, to create medical support systems for a human being to be away for such a long time. I believe that when this project is completed, we'll have more questions than the answers. Although right now, many scientists think that we have received some interesting results. But this is a research project. This is the project that is needed to make sure we make minimum mistakes when we work on specific planning and programs. That's why we will not spare our efforts. Thank you very much.
I can't help asking myself what would motivate somebody to volunteer to shut themselves up for 520 days. I mean, it's almost like volunteering to go to prison for a year and a half. But seriously, if you're studying the psychological impact of a potential Martian crew, I think the motivation for why somebody would participate in that has to be very important. And I wonder if you can comment on what are the motivations that you perceive causes people to be willing to give up this much of their lives to do this project?
I have to know that people can be confined in prison for a longer time, and some people, even if they did not commit a crime. As to motivation, of course, the motivation is there. I am a cosmonaut myself, and I have been working with cosmonauts all my life. I have a lot of friends who are dealing with diving and go to the mountains for a long time. And this is a trait of their character, that they have to be curious, healthy, curious people. And also they have to be willing to learn something new, and there is something to find out something new and to learn something. This is not just confinement in our experiment. This is not a punishment. We have two people who are collecting actively data for their science program, for their own projects during the confinement. This is a motivation for them. Of course, they have a lot of support, especially in Europe. There is a lot of attention being paid to their science program. Also, what other motivation is, they have to test themselves, and this is what they're doing. Sometimes I'm surprised myself, but you can find people like that.
It might be best if we just had you talk with Dr. Markov, and we'll get on with the one brief thing.
Sure, I'll try and make this quick. Dr. Markov, I think we all applaud the research effort, and I think it's going to be groundbreaking. And I fear it's going to shed light on some of the extreme challenges and difficulties of sending humans on a historic mission to Mars. Just 2 points. One is, even with the 520-day study, we'll still be 13.5 months short of a Mars mission, so I think the results will be very important. Just one other comment on self-monitoring and performance, because you alluded to, people's actions and how they respond to other individuals. One thing I think we've noted in the Antarctic in long duration is a slow deterioration in psychological status. And there's a loss of objectivity. There are no external references. And I wonder if some monitoring from the ground in terms of how crews are performing individually and reacting to one another in terms of, voice layered analysis, heart rate variability, and also some of the work that Dr. Dinges is doing on facial recognition and emotion, and getting some of that data back to the ground so there can be some outside objective monitoring of what's happening with the crew, and that those objective observers can suggest increases in countermeasures and that type of thing, because my experience has been that there's a slow deterioration during a long duration mission, and individuals have difficulty recognizing that. Thank you.
We have multifaceted psychological observation. Individual qualities of the crew members are being observed as well. We know that in space, sooner or later, we observe some psychological changes in people. And productivity, ability to act in general is also another thing that will change. Interaction between crew members, changes between the crew and the support crew on the ground. Psychological and emotional sphere is the subject of observation. Also, we have several experiments devoted to sleep of the crew. What I didn't mention also, we intentionally complicated the situation for the test subjects. Every day for 24 hours, one of the crew members is on guard, and he's supposed to communicate with the support crew if necessary, and this guard is supposed to watch the systems of the module. So the research is comprehensive. Out of 70 experiments, 30 of them are psychological type, and many of them are also comprehensive, and 3 of them are American projects.
Thank you very much, Dr. Markov. That was an excellent presentation, and I'm sure everyone has a lot of interesting questions, and perhaps if you would give them time later today to meet with them,
I'm ready.
Talk with them. Good. And thank you. That was excellent. It's an outstanding project, it seems. Today we have something a little unusual for the summit. There are actually 2 novel presentations in the sense of new discoveries. One was brought to our attention by Dr. Tom Caskey, who is a professor at the University of Texas, a very well-known geneticist. And it's from one of his students of years past, who's now a professor at the University of Texas, and it's Dr. Ching-Chi Lee. And he's going to talk to you about induction of a hypometabolic state in mammals. Ching-Chi?
Thank you. I'm Ching-Chi Lee from the University of Texas Health Science Center across the street. I'd like to thank Tom Caskey and Bobby Alford for making this possible to speak to such a distinguished audience. I think most of you who in this field recognize this slide that came from a Star Trek movie. I think if we can achieve this process, then the trip to Mars could be a lot simplified. Right. So can I? Okay. So I think by and large, can I get this thing going? Can I get this thing going?
One second, I got to switch over.
Okay. So I started this project 6-7 years ago. My interest is in circadian biology, but I also have very deep interest in the process of metabolic control. And this is a slide of a hibernating animal, just to sort of illustrate the point. If you keep an alpine mammoth when they go into hibernation, what you notice is that the core body temperature of this animal could drop rapidly towards close to ambient temperature. What is very distinctive is that the oxygen consumption drops very rapidly, just parallel to the drop in core body temperature. You can see this very, very rapid drop. And then, as you can see, over time, as the core body temperature drops, the amount of oxygen needs is dropped dramatically into about 5 to 10% of what an animal would need. Right. So when I started this project, I wanted to get into track this by using genetic tools so that we can find a gene that we can use it as a marker to find the molecular basis of how this thing could work. So we basically started out simply by looking at a non-hibernating animal. I say if we can find it in a non-hibernating animal, then perhaps we can do this in human. If we find it in a hibernating animal, then basically we may be just replicating what nature is doing that may not be useful for human beings. So what we did was we chose a mouse. Mouse are non-hibernating animals. So we kept a mouse in constant darkness and one that kept in light dark cycle. And then we took the liver from this animal and get mRNA from it and asked the question, can we find genes that are differentially expressed? And lo and behold, we found one here, that is this gene over here. And this gene is not very particularly unique. It's an enzyme that encodes for breakdown of co-lipase, and its job is a partner for pancreatic acyl lipases, and its job is to break down dietary fat into fatty acid. But what is unique about this gene is that because of its role in breaking down dietary fat into fatty acid, it is strictly expressed in the pancreas and in the stomach, okay, and nowhere else. So as you remember, we did a study by the microarray looking for genes that are expressed in the liver. So there was this dichotomy that we found. So we said, well, maybe the microarray is a mistake, and there's something wrong with it. So we went ahead and did another method of analysis. This is true northern blot analysis, and asked ourselves, now is this gene expressed in the liver in the light dark cycle? And lo and behold, what you find is that in these animals, that this lipase gene is not expressed in the light dark cycle. But if you keep the animals in constant dark, you can see this gene is expressed. And that basically told us that, hey, we have a gene that's turned on just by strictly keeping the animal in constant darkness. And here you look at the tissues in light dark cycle and in dark cycle, and you can see that the genes are expressed in most of the tissue except in the brain and in the kidney. Very unique. So the question I want to know was, what is the signal that regulates this gene expression? I don't want to study co-lipase function because that is well known. So what is the key question is, what is the signal? If I can identify the signal, then I can track into the problem of how we can look at this metabolic control. So this is a 4 years project of trying to identify this molecule. And sure enough, we kept animal in light dark cycle. This is morning, evening. And this is a dark cycle, morning, evening. And if you look at, this is HPLC. Looking at this peak here, number 2, you can see that there is an oscillation of this peak number 2, and also there is an increase in the DD cycle animal. So we found out that based on spectral absorption, it was absorbing very highly at 216 nanometer. Straight away, we realized that it must be a nucleoside or a nucleotide. Right? So basically what I did was we asked my postdoc, go ahead, go to the catalog, get all the nucleoside and nucleotide, run it on the HPLC, see which one would match precisely with these at 8.5 minutes. Right? Lo and behold, he ran it, and here you are. My heart really dropped when he told me what it was, and he told me that the peak, the nucleotide that matched perfectly was 5' AMP, which is essentially the breakdown of ATP. Right? ATP, ADP, AMP, the last product. So sure enough, we went ahead and did it with some nucleotide, actonucleotidases to remove the phosphate group to convert it into adenosine, and sure enough, we found that this was the case. But then we got this stuff from Sigma and said, well, if it's AMP, let's inject it into the animal and see whether the genes will be turned back on in this animal that kept in light dark cycle. Sure enough, you can see in the saline animal, the co-lipase is not expressed. This is by RT-PCR. But if you give 5' AMP, the gene for co-lipase is induced everywhere. So we really have the right molecule, right? But then my postdoc, being from China at that time, he doesn't use forceps to pick tissue up. He uses his finger, right? After he inject the animal, he took his finger and pick it up. And he told me, well, the organs are kind of cold. I said, are you sure? He said, yeah, I'm sure, they're cold. So I said, well, why don't you put a probe into the backside of this animal and see what temperature they are? And he told me that they were basically about 1 or 2 degrees above ambient. In this case, 26, 27 degrees after injecting the AMP. I was astounded. I said, wow, 10 degrees difference. There's no possibility of this. I said, is the animal dead? No, he said the animals are alive. So here you are. So here we are. We have a little video. Just a little quick one. So these are more little stuff now. Those are published stuff. So these are just black 6 mouse, regular laboratory mouse. Okay, give AMP. We kept them at 15 degrees, room temperature. And the reason we are picking them up is to illustrate to you there's no anesthesia involved. In other words, these animals are basically in deep hypothermia. You can see they're still responsive. They still move, you see? They kind of, see? They do move, okay? Right? So basically, they are in a deep hypothermic state. So the core body temperature of this animal are roughly about 16 degrees. All right? Okay? I'll be very quick with this. Just one more, another 10, 15 seconds. I'll move on. Okay, just watch this animal a little bit. You can see the reason we left it on is to show you that these are not, and there's no effect of anesthesia, right? If you have anesthesia, you're basically in a comatose state, right? And in addition, if you use anesthesia to cool, you cannot cool it down to this level. You try anesthesia on mouse, probably 22, 24, and they basically there's no return. And in this case, we can keep it at 15 degrees core body temperature. So because of time, I'll move on and describe a little bit to you about the inside of this process. So here we are. This is a normal heart rate of a mouse at about 37 degrees core body temperature. This is the same mouse when it's cooled about 16 degrees. You can see the heart rate is about 50 beats. In other words, 1/10 the rate, right? And in addition, you can see this is the heart rate, and this is the breathing rate. So the breathing rate can also be reduced dramatically. So in other words, not just only the body cools, but the physiological processes that accompanies also slows, right? Now you can see now the demand for oxygen demand, the production of carbon dioxide will all basically slow, right? Your need is dramatically reduced, right? Okay. So what is the interaction with this? So these are all animals given. There are 3 animals given AMP and 1 animal given saline. You can see the saline animal do not cool at all at room temperature, right? But if you give AMP, what you find is that they all display this very rapid drop in oxygen consumption, and then followed by this second phase that corresponds directly with the decline in core body temperature. Now, what is critical here is the temperature of the room, right? These are both same dosage of AMP, right? But what you see is that if you keep the room at 23 degrees, the lowest it will drop to is about 1,000. Now, on the other hand, if you keep the room at 15 degrees, which is 60 degrees Fahrenheit, the lowest its drop will be about 300. Right? It's a huge difference, 3-fold difference, roughly, right? And not only that, what you find is that the drop in core body temperature will drop to about 25, 26 degrees. In this case, it will drop to about 15. Huge difference, right? Just by changing the temperature of the same dosage of AMP. But what is astounding also is that how long it remains. Here we're talking of hours, here we're talking of minutes. If you keep it warm, the animal comes up very quickly. If you keep it cool, it stays down pretty long. In this case, up to 9 hours, right? So if you don't do anything, about 9 hours is the boundary, right? Now, you cannot stop the animal from coming out by giving it extra AMP or by keeping the temperature lower. If you do that, it's fatal to the animal, right? Now, if you give AMP, extra AMP, it's also fatal to the animal. In other words, once aroused, you cannot stop it, right? So that was the challenge I have, is how can we make this thing go much longer, right? It turned out that to control this process is what we are working on right now, right? I can't tell you the exact cocktail of it, but we have made considerable progress, okay? So this is the latest, because I'm basically reverse engineering the whole process. So we did an entire metabolomics analysis of the blood in the system and asked the question, can we find what are the changes in metabolites in the process, and then pull the metabolite that is altered and see whether we can get back into the animal and see whether we can alter this length of hypothermic state, right? And as you can see, it's a whole bunch of cocktails, so I cannot tell you what are the exact mix of this thing, but I do have some ideas what they are. And there are 3 animals here that came out before their time out, but you can see they're all way beyond 9 hours. In this case, 24 hours. Here we stop it because we increase the temperature to 22 degrees, and we induces the process to come back up, right? The idea here is to work out what is this cocktail that we want to have that will allow us to do this thing, right? If you inject AMP alone, trying to suppress this, it's fatal. So in other words, this is where the boundary is roughly about 12 to 14 hours. That's where it is. So in other words, these are all natural metabolites we're working through. I cannot tell you the exact combination because we're still reverse engineering the whole process and see which one is the essential ones, okay? So basically, I'm very hopeful right now that we can extend this process to where this is 24 hours, and I think we can go beyond 24 hours, right? And for a mouse, that's phenomenal, right? I think for a bigger animal, it's going to be a lot easier than to deal with a mouse. So here we inject periodically with this cocktail. This is where we're injecting, right? So to end this, we can show that this thing is not only applicable to mouse, we did that with a dog, and it also worked. And then we did one monkey with a collaborator in China, and showing that this process, if you give to a monkey, it also responds the same way to AMP, the fact that you can cool it. Right? In other words, it gave you AMP, you can say between half an hour, the body temperature 35, and then at 1 hour, 27. And then we stop this process, and then we can let it go back up to normal. In other words, the basic biochemical process, the induction, is likely preserved all the way to human. That's the take-home message, right? So we were using the mouse as a way to work out all the the hard work of how this thing comes about. All right? And I thank you for your time, and I'll be happy to take any questions. And these are the people who are in Walmart Lab. Okay.
The same thing, Chenchi. Lee, thank you for bringing that new discovery to us. I'll leave it for the rest of the morning for people to ask questions. Yeah, for a flight home. Okay. Well, no, they're not any questions. The next presentation, in the same vein but in a different subject, is by Professor Tour, who's a professor of chemistry and quite a number of other things at Rice University. And last year, you may remember that he spoke to this group during the time of the radiation panel that we had. And so, Jim, is he? There you are. Yes.
Thank you. I'm not a physician, so I'm sure I can't answer a lot of your questions, but I'll tell you some of the things that we've found in our research group. And it turns out that, as many of you might know, there was a long series of tests that were done from 1959 into the early to mid-70s on protectants and mitigators for the effects of radiation exposure. It's interesting that this topic is being reinvestigated, not just because of space travel, but more so because of the effects of what might happen here on Earth due to terrorist attacks, what might happen if a nuclear reaction were to go off in a city and you had 1 million people exposed or soon to be exposed. Is there something that could be done to protect or mitigate the effects of this? Some of the experiments that we've looked at have looked at nanometer-sized particles for delivering different agents, both IV and IP, as well as orally. And if we're going to have a radiation event here on Earth, and there's a large number of people exposed in a city, say 1 million people, there's no way that an IV could be administered. Really, the only thing that we would have is to give an oral dosage to massive amounts of people. And the Department of Defense has studied this, where there would be dosages that would be placed in supermarkets, and people would go to the local supermarket, get their dosage, and take it right away, because you can't get to doctors' offices, you can't get to hospitals. And you can't just say, well, if you're feeling sick, then take this. Everybody will be feeling sick. Everybody will want this dosage for themselves and for their families. So really what we have to do is develop something that is oral, something that could be taken, and even if it's not a mitigator, in other words, even if it does not work after exposure, being able to work before exposure is also could be extremely effective in a place where there's a lot of exposure, in the sense that what happens is the radiation travels as a plume, and people can have minutes to days knowing that the radiation is coming. And you would say, well, why don't they just get out of the way of it? They can't. It goes out in a large vector, and it covers a large swath of area. We've seen even in this city that trying to get out with 4 or 5 days notice that a hurricane is coming is almost impossible. So when you have a large city, the exiting the city, even when the infrastructure is working, is impossible. So can we have something? And so what we've done is we've looked with, this is work with the work of Luca Millis and Kathy Mason at MD Anderson Cancer Center. And we've used the traditional study of studying the crypts in the mouse jejunum to look at the effects of the stem cells in the crypts. And this is an assay that's been used since the 1970s, and in fact was developed in collaboration with MD Anderson. And generally what we do is we dose with 12 grays of radiation, 12 gray of radiation, which is a fatal dosage. Typically the mouse would die in a few weeks from that dosage. And we study the effects for 3.5 days. They're allowed to live 3.5 days after the exposure. And then we count the crypts in the jejunum. in a slice of the jejunum. Usually there's about 150 to 160 crypts in a normal mouse. And so what happens, for those of you who are less familiar with this technique, is that once the stem cells in the crypts are gone, the stem cells generate the cells that line the villi. And when those cells stop being made, what happens is death by GI syndrome. So there's bacterial infections that occur without that layering that covers the small intestines generally. And that's the main source of death upon the radiation exposure, is death because of GI syndrome. And so can we protect that layer? We have found oral dosages of drugs that can be administered 30 minutes before the radiation event and have, generally, if there's no treatment, you'll have about 10% of the crypts remaining. So 10 to 15 of the starting 150 to 160 crypts would remain. We're now seeing over 2/3 of the crypts are able to remain by having a single oral dose 30 minutes before the radiation. It turns out that compounds not terribly unlike this and quite related were studied in this series that were studied back in the 1960s. And further studies were done, and what worked on mice didn't translate into larger mammals. But what we're seeing is that because we're looking at a community which is potentially much larger than the community of people that are going to be going on space travel, that we can leverage a lot of funding from what now would be the Department of Defense and DITRA and agencies that would want to protect large numbers of people and the National Institutes of Health to try to attack this problem once again. Do we have something that would allow us to take this into larger mammals, move this effect into larger mammals? There are a number of compounds that we in particular would like to look at, would like to continue to study this work. But again, it's in small bursts of support. To give you an idea, we had a DARPA grant that was funded. They said the program would only be 12 months. Well, if you know what 12 months of research can do in academics, you're just waiting for the students to arrive. And then when we got the grant, they asked for the timeline to be cut back to 6 months because they wanted a solution. And they said you can use the same 12-month amount of money, but spend it in 6 months and find a solution. Well, we finally agreed upon 9 months. The 9 months ran out. We were I think, making very good headway with this oral dosage that's able to protect 2/3 of the crypts. But again, the funding ran out because it's typical type of funding that comes with effects that we want to get a quick and fast solution to this. But many of you know there's no easy solution to this. But I think that we are showing that we're within reach, and if we reinvestigate something that has been investigated 40 years ago, I think that we can really crack this problem. We have nanometer-sized delivery vehicles. We have nanometer-sized particles that can go in, and things that could potentially sequester far more radicals. And this is the course of what happens. So initially the radiation would cleave DNA, but if there's a cascade of radicals that ensue from these events that cause further radical damage to occur. Also, if we can have cell arrest to occur to allow the DNA to be mended before the cell goes into apoptosis. So there's a number of mechanisms that could be discovered, but I think that we are within reach of finding an oral cure to this, or at least a protection, if not a mitigator as well, by studying a series of strong radical traps that could be administered. And in fact, many of the compounds that we're looking at are so non-toxic that people could take a daily dose day after day with no effects. And we've tested these IV, IP, and orally. And many of them actually are compounds that have been known on Earth for a long time and studied for other sorts of other studies that they've been looked at. And so people could indeed take large amounts over a large period of time.
Thank you very much. I realize these were compressed presentations, and at the same time, they were important. They're important bits of information to bring to your attention and hopefully stimulate your interest in some of those particular discoveries.